Okay. All right. So, hello everyone. Let me close at least this door. So, thank you all for coming. You should all in your email have received a link to this page. Okay. So this page, we'll talk a little bit more about this page, but this page will have all of the links and all of the documentation and everything that we're going to use for the next 3 hours. And as far as I'm concerned, this page will stay up indefinitely. So you don't have to worry about saving it. You can save it if you want, but we'll probably be updating it during the workshop. So just some sort of prefatory materials. The schedule for, sorry, I'm on the wrong, on the wrong window. Okay. The schedule for today is roughly, people can see that? Okay. The schedule for today is roughly between now and a break, we'll do Shell and some RStudio. During the break, we're going to run something that takes like 10 minutes, so that we might need for the end. And then afterwards, we'll do some more RStudio stuff and some R Markdown stuff. And forgive me for the vagueness. There's actually a lot of materials and stuff below that's less vague, but okay. So just from the introduction perspective, so I'm Titus. I'm a professor here in Vet Med. I'm also teaching the lab section for the Genetics Grad Group 201B course, which is I think something like Intro to Bioinformatics. I don't know what the name is. And as part of that, I'm offering a couple workshops. So far, we've done one Shell workshop and then this workshop for people in the class who don't necessarily have a lot of computational background or want a refresher. And since offering a 3-hour thing for like 5 people is slightly demoralizing, this week I opened it up to more people. So sorry for the short notice, but I do everything at the last minute anyway. So so in the room we have 2 other people from my group. In the front is Taylor Ryder. She is a grad student in food science and technology. Okay, it is and technology. Food science and technology. Don't worry about it. She actually knows a lot more R than I do. She actually likes R, which I don't particularly. I use a different language called Python most of the time. But a lot of the workflow and stuff that we're going to talk about today is the same in R and Python. We're just going to be using R because RStudio, it's basically, it's more intuitive, it's easier, it's better, and all these other things. So so I've asked Taylor to tell me when I'm saying something incorrect or incomplete, and she may, depending on how she feels about the job I'm doing, she may teach some or all of this after break bit. Also just to break things up. In the back we have Karen Word. Karen likes to sit, observe, and then tell me why I'm doing a bad job teaching. That's actually her job in my lab as well. She's involved with evaluation and assessment of training and workshops and stuff. So if you have any thoughts on other workshops you'd like to see or additional materials or so on, ask Karen and or talk to Karen at the break or send her feedback. And she may or may not talk to you. I don't know. Her feedback. All right. I mean, she will be happy to talk to you. She may seek you out at the break if you're standing around looking for it. Okay. A couple other things. We're recording. What that means is there's a camera, there's a mic somewhere, we think on the computer here. And this is going to YouTube as a non-public link, which means it's not findable or anything, and we'll send you the link afterwards. The main purpose of that is not necessarily, I don't necessarily feel like people will like find the link and then watch it and then learn all this stuff. but a lot of the value is actually the doing it and having the help here. But the link will be for you all to look back and like, I didn't get this point. What did he actually say? What did he actually do? So, and you can do whatever you want with that link. If you want to send it on to other people, that's fine. So from that perspective, if you ask a question in a loud voice and identify yourself with your social security number, that may end up on a YouTube recording. So, you know, just keep that in mind. We can scrub things from the YouTube recording. I can't imagine why we would want to do that, but if we do, if you want something removed, that's totally fine. Sticky notes. We have not yet handed out the sticky notes. It's right here. Thank you. We're going to be using sticky notes to paste things and to flag for questions. We have some more. At least one more of these, by the way. Plug in. And there's things on the ground. Sticky notes. We're going to use sticky notes to paste things. So what we're going to do is with that is we're going to when there's a task or a question or something, we're going to ask you to take all the sticky notes off the back of your laptop, and then when you're done with the task, we'll ask you to put the green sticky note up. And that way we'll know that you're done, and we can sort of suss out how far along the room is. The red sticky notes are if you have a question and you don't want to hold your hand up, and I'm ignoring you, just put the red sticky note up, and either Taylor or Karen will come around and here should work. Here should work if you just beware. That's a camera, so okay. That's fine. Thank you. So what did I want to say about that? So we'll work through the sticky notes. Sometimes I use them better than other times. If at any point you have a question or a problem, put up a red sticky note and someone will come find you. Okay, I've already said this. These materials will stay up. The links below will stay up. The binder, which I'll introduce you to in a moment, will stay up. And the main thing that I want to say is you can actually edit this page. So if you go up to the top, there's a split pane view and a full pane edit view. And if you go to the split pane view and you scroll down, you should be able to, sorry, I can't see my cursor. You should be able to, let me just do this. You should be able to add stuff. And so this could be, if you have questions that you want to write out, or there are notes that I make a comment and you want to put a note in, or say, hey, could you add a link in? I can put a link in here. You can put a link in here. And then they'll stay on this form and they'll be available for everybody else. So one place where this comes in handy is if I've screwed up the materials, which are mostly going to be copy paste. If I've screwed that up, which happens, I can easily put a corrected thing here for you to work with. And if you're not familiar with HackMD or Markdown, it's a super great way to take notes, collaborative notes during things. So, okay. It updates, it live updates and all that other stuff. Okay. So let's see. Sorry, I need to just a little bit further. Okay. Bathrooms are behind us. Oh, sorry. For the materials, you can send the materials to anybody you want. Don't worry about it. They're all essentially free and clear. Bathrooms are behind me at any rate. Behind you is the atrium. Behind me there are two bathrooms up there, and there's filtered water in the kitchen over there. There's another bathroom way down the hallway. And please feel free to just get up and do what you need. There are cookies over there. As I said, there's water. Sorry, there's no coffee. The Scrubs coffee shop during break is sort of on just across the street over there. And they have a, what kind of coffee shop is it?
Pete's.
Is it a Pete's? Okay, they have a Pete's there. They also have all sorts of food stuff. So my goal is to, in terms of the schedule, my goal is to have plenty of time for a break, make plenty of time for a break. If we don't get through all the materials, that's fine. And what I'm hoping to do is have this be paced to the questions and the progress of things. Sometimes I go a little bit slow for people that already know some R. very little of the R that we show you is going to be high-end, super cool, technical stuff. A lot of it's going to be how I think about data analysis in the context of bioinformatics, and how I use even very simple R commands to get a sense of what's in the data, and then try and tie that back to, well, ultimately biology, but I guess genomics would be the sort of, depends on whether, I assume genomics is biology, but, and so the process and the discussion, please feel free to ask questions about what we're doing. If you have super advanced technical R questions, Taylor can answer them. So please go ahead and ask, or at the worst we have Google. So, and that's how all of us figure things out. So we'll just show you how to do, advanced Googling skills, which basically involves asking the same question, asking the question 3 different ways and finding which one actually has the answer that meets your needs. Okay. Okay. So any questions at this point? So let's see, what do I want to do? Okay, somebody remind me, right before, what does BLAST do? We need to click on this link, but don't do it right now, because it's going to take a couple minutes to start up. So, what we're going to talk about today is, let's get this right, it's, there we are. There we are. Okay. Get a nice blank screen for everything. Where did the mark? Oh, here. I'll leave these markers here for others. Okay. So what we're going to talk about today is the way I almost always do my bioinformatics. So let me take a step back, since there are a bunch of people here. I don't recognize everybody in the room. So so I'll do like a short introduction of what my lab does and what I do. So and I'll keep it short, so for those of you that have heard it before. So I'm mostly interested in, so my lab is called the Lab for Data Intensive Biology. And the idea behind it is that as gathering data, in particular sequencing data, becomes easier and cheaper, people have more and more data, and a lot of biology can be, some of sort of biological research practice is being moved more into the computational realm, where you can do hypothesis generation and hypothesis development in a purely computational sense, and even do hypothesis falsification by looking at public data sets and exploring, or data sets that a previous person generated, and exploring your hypotheses using data that was maybe generated for a different question. So my lab is purely computational at this point. We have some people in the lab that do experiments. Taylor does experiments. Mostly at the moment, DNA and RNA extraction, and then also olive oil, something characterization, right? Lots of stuff. I have other people in the lab that either do or do experiments or collaborate closely with others. But the main focus of my lab is thinking about methodologies and how to do a better job on the computational side. And a large part of that involves running lots of programs on the data in a series of steps, which we call a workflow, which is generally called a workflow. The idea of a workflow is you have some data that you generated, and you have some step that takes the data and filters it or cleans it up, and you have another step so you typically something like cleaning, and then what I'm gonna make up a word, boil down analysis, which is to say you have some large amount of data, and you suppose you're doing variant calling a pile of reads, and you align them to the genome, and then you get out all the variants that are completely alignment. And here you go from a large amount of data down to a small amount of data. And then you do some kind of, and these steps, like, there could be many more steps in this. I'm just sort of outlining the general idea. And this step 3 could be statistics, exploration, or graphing. And we're mostly going to talk today about graphing, just because I really like looking at data. And there could be many more steps. And the trick is really that for these workflows is that you can do all of this stuff sort of manually. What I mean by manually is you can either do this stuff on your laptop sometimes, or one command at a time on a server, or using a graphical interface. But these steps in particular take a lot of time. For variant calling for the human genome, these steps can take a week or two, for example, if you're working with a large genome. And then the statistics and the exploration of the graphing, because you've now gotten down to sort of a smaller data set size, these can actually be done interactively. So, I'm going to say, before you get the data, if you're not using public data, is some sort of wet bench work where you're generating the stuff to be sequenced in the case of sequencing. Step one is transferring the data around and cleaning it. Slow and needs a big computer. And then this is also slow. And then at this point, this is the exciting and fast bit. Because now you've done all of the sort of boiled down analysis, you have the smaller set of data, and now you can really explore your data set and see what biology is in there. And we're going to talk a little bit about this in the context of something called BLAST, which I'm sure all of you have run at some point or another. We're going to go into maybe some detail depending on what kind of questions and what kind of eye contact I get. Generally, I find if people aren't looking at me and nodding, and they're just staring at me, then I worry and I slow down. And if they're checking Facebook, then I know they're either bored or they've lost track. So it's okay if you're checking Facebook because you know all of it. Sometimes because you know everything I'm talking about, but sometimes we also step off a cliff where it's like, well, I knew everything you said up until this word, and now feel free to ask questions to re-engage. I'm not gonna I just said that 30 seconds ago. By the time, if I repeat it like 5 times, then maybe a lot of chat. But and so our goal is to sort of get here. And the thing that I like about this is that this is where you sort of reach a joint understanding. There's the data itself, how you generated the data, the experimental design, and the way that what is actually in the data. And then there's the biological purpose that you want to put it to. And this is sort of like the whole point of why you're going through the rest of the stuff, right? I really I'm a little bit weird. I'm very weird. I try to hold off on saying anything about the biology until after I've understood sort of what's in the data, independent of the biology, why the data looks the way it does. You'll see some examples of that in the workflow. And then once you're like, you know what, I think I understand what's in the data, now let's actually start generating our hypotheses, testing our hypotheses, thinking about our specific biological hypotheses. And I'll just say that faculty tend not to want to know about anything before step 4 in this fictitious workflow, which is hypothesis. Well, let's say write-up is probably a little bit strong, but I think I know what's going on. Here's what my actual hypothesis is, and then they can tear you to shreds at that point. But before really explore the hypothesis, ask what other techniques you can do, what other literature supports it, that kind of thing. And before this point, there tends to be sort of a disengagement with a lot of bio faculty where they're like, well, bring it to me when you have some graphs. It's sort of the attitude. Bring me the results when you have some graphs. We're not going to talk in this workshop about how these steps can really affect the rest of your workflow. But of course, it's just like everything else. If you mess up your wet bench experiments, mess up your data cleaning, mess up your follow-on analysis, then it doesn't matter how nice your graphs look here. You're just standard warning that I just want to put out there. Okay. So are there any questions at this point about workflows? Okay. The last point, and please feel free to ask. Anyway, the last point is these. I should maybe use a different color. Let me use purple. These are typically executed at the shell, and there's a bunch of reasons for that I won't go into. If you don't know what the shell is, we'll have a brief introduction to it, but I did a workshop on it last week. And the point is that these can be done on remote servers. These can be done in massive throughput, which is say I just got 100 gigabytes of data, and I just want to do all the cleaning and all the boiled down analysis on all 100 gigabytes of data. And that may take 3 weeks. I want to run the commands, and then I want to walk away and do my other stuff. This almost always now takes place in what I'm going to call a notebook environment. And today we're going to be using RStudio. How many of you have used RStudio before? Excellent. Okay, so then you already sort of know what I'm talking about. It's an environment where you can interact with the data, see plots, explore the data, and save some of what you're doing. What I'm sort of most interested in today is sort of connecting the shell to the notebook, showing you how you might use the notebook, and then talking about how you can make this notebook environment, how you can document what you've done in the notebook environment so that you and others in 6 months can figure out what you did. And maybe do the same thing to your next data set. And for that, we'll be using R Markdown. How many of you have run across R Markdown before? Excellent. Okay, great. So you'll get a chance to see it. It's very straightforward and follows directly on from RStudio. There's no magic. It's just putting the commands in a place where everything gets tied together. Okay, so what else did I want to cover here? Sorry, I have to go find my. Okay. Just to check, everybody has access to this set of materials. Yeah. Okay. It should be in your email if you haven't. Okay. So before we move on to what does BLAST do? Before we move on to what does BLAST do, which is I'm going to just talk about to make sure we're on the same page. On the page, there's this thing that says Launch RStudio Server. And you can open that in a new tab, or just, or you can click on it and then open a new thing. And you should see something like this, Binder, and it's going to be running, and that's going to take a minute or 2 or 3, and hopefully it'll work. Because if it doesn't work, we're going to have a really boring workshop.
Question.
Yes.
So I was wondering, when we are going through this whole step process from 1 to 4, it seems like an extensive process. So there's also checkpoints in that?
Yeah.
Or you just like go all the way and then found like you just messed up?
So you are asking an excellent question. And let me answer that excellent question. Okay. Oh, mine came up really quickly. So that's good. So so the question, just to make sure I understand, the question is, there are checkpoints in here where you could say, does my data look good?
Yeah.
Does my boil down analysis look good? Do my variants look good? Do my statistics, do my plots look reasonable? Is there significance? So there's a lot to be said about this, and I'm going to give you a slightly maybe contrarian perspective, which is, for me, I'm really endorphin driven, which just means like I want to get somewhere, and I found that getting to the point where I have plots is exciting, even if plots turn out to be wrong. So what I do is I do these steps and I keep track of what I'm doing. I either automate it, I record what I'm doing in a shell script, or I automate it using a workflow system, or I just make the equivalent of like a HackMD document or a Word document that contains all my commands. And then I get here, and then I look at the graphs, I'm like, is that even remotely reasonable? And at that point, I can go back and I can look at these checkpoints. But I've gotten to the point where I have something. Tangible is also a weird word to use for computational research, but I have something in front of me, and I know how I got there. And if I have to pack up and go home for the day, I can recover where I am here much more easily than if I'm halfway through figuring out this analysis. So the reason that's contrarian is because if you think about the way you run controls in Webbench work, you would, for damn sure, run a gel.
That's true. I'm just off over here.
Right? Yeah. And the difference there is, if you had a robot, you might not actually run a gel between all the steps, because you could say, you know what, just do all of these steps, and at the end, if it didn't work, well, I blew some reagents and whatever, but I have the robot worked out, so now I can just like robot's workflow worked out, so now I can go back and say, change the PCR, whatever, regenerate the data. I don't know. It depends on how much each step costs in terms of time and resources. If this is going to be a costly step, if this step is bad, then by all means stop and checkpoint. If this is going to be a step that consumes a couple days of computer time but is otherwise not a big deal, I would vote for going all the way through. The other thought that I had is, there's a tendency for people to get really caught up. You're like, oh, if I'm going to do variant calling, I'm going to use 8 different mappers against 2 different versions of the reference genome. I'm going to explore all the options. And that means that like 2 years into your project, you're still trying to get past this step because you're exploring all the options. Whereas, and this is where I become kind of cynical and nihilistic, so I apologize, but in reality, it seems really controllable because it's all in the computer and reproducible and you're learning new stuff often and it's exciting. The odds are pretty good that if you messed up in your experimental design or your data collection. And so spending a huge amount of time optimizing this before you get to the point where you have any idea if your hypotheses are good, maybe a sidetrack. I've noticed this happen a lot. That's why I sort of like get to the point where you have an answer. If the answer looks like, if the answer is like, there's some interesting genes in there that have been published, and I'm making, if it's an RNA-seq workflow, there's some interesting genes in there that are in the literature. Now what I want to do is fine-tune the statistics, fine-tune the sensitivity, fine-tune the specificity. You can go back and start fine-tuning this in the light of this. That's dangerous because now that's also dangerous. It's kind of like p-hacking because now what you're doing is you're saying, based on my results, I'm going to improve my data processing pipeline. So it's something else to keep in mind. But with large-scale data analysis, it's actually surprisingly hard at these stages. This is all one reason I don't want to make these videos public is maybe I get a lot of flack for this, but it's actually rather hard to bias these steps. Usually what you do is you like eliminate too much data because you're being too stringent with cleaning it up, or you don't eliminate enough. And so you get either a lot of noise or not enough signal. And so that's really with these what I think you're fine tuning. Is that a fair? Yeah. So I don't know. So it's a philosophy. We're still working out how to do all of this. At the end of the day, I would generally suggest not generating a hypothesis solely from high throughput sequencing data. I would not suggest evaluating a hypothesis solely from high throughput sequencing data unless you are in a lab that does a lot of statistics. Generally, what I would say is this is now you have a hypothesis and now you're going to go confirm it some other way, either with targeted experiments or with other kinds of analyses. Because with anything, it's too easy to fool yourself. Right? This is still only one line of evidence, is basically what I'm saying. So seek other lines of evidence. And then it doesn't matter if you p-hack, sort of, because at the end of the day, you're going to validate this some other way. That would be my hope. Question?
Our studio using that download for your desktop, but there's like a server version too that you have to pay for. What's this one?
This is a server version that you don't have to, that is free for us to use. Yeah. No, I don't actually know. It's free for everybody. Do you know what the RStudio server situation is?
It's the cloud one that just launched.
Yeah.
It's brand new and free for everyone.
No, the binder one. RStudio server. I think it's free for academic use. You know what? Let us use the Google.
I mean, it's free in quotes, right? Because
Is RStudio server free to use? So it is actually free for use. It's free for use if you pay for support. Sorry, now I remember. So you can use it. If you want to install it on a central campus server, it may cost money, but it's free for us to use in this model, just like RStudio is.
is there a difference between desktop and the like online version, like in terms of what it can handle?
I don't 100% know all of the answers to that. I will say that the server version can run on bigger computers than your laptop.
But RStudio in general is far more buggy than our kernel, and it's oftentimes if you're running a large process, it's going to time out even if you throw a lot of compute at it.
Okay, so basically, RStudio itself is robust. It's underlying R that has problems, that may have problems with big compute.
Opposite.
RStudio is buggy or R is buggy?
RStudio is buggy when you throw something big at it.
Okay.
It won't necessarily, like it'll stop, but it won't give you the error message.
It'll just.
If it's huge, and like, but that's when you get too really big.
So what's interesting about what Taylor's saying is this actually ties into the workflow pretty well, because these are things that are slow and require big compute. And these steps, if you do them in R, you can actually do them at the command line where you don't need to have RStudio running. You can do your big your big compute at the command line, and then import the results into RStudio to visualize. So these notebook environments are really for visualization and exploration, and then the shell and stuff is really for executing these big long-run jobs. How many of you have access to the Genome Center cluster or the farm cluster? Anyone? So then, do you have access? Are there big computers that you have access to elsewhere anyway?
Wait, are you guys all in GGG or IGG? Or are you in the College of Ag and Engineering, CAES?
I'm in IGG.
Because if you're in CAES, you have farm access.
You can get farm access.
Right, like, but.
Yeah. Okay. Well, so we can talk. I'd be interested in knowing where you would be able to do big compute if you need to do this for like next-gen sequencing data stuff. We can talk. And I have suggestions and stuff that's. But for today, we don't actually need any of that. We're just going to use this binder thing that we're running. Okay. So let's just keep binder awake. If you have this up and running, you should have a tab that says RStudio. Just hit enter a few times at this prompt, and we're trying to keep the computer from going to sleep. If you just got here, you should have an email with a link, and just ask the person next to you what to click on. Or flag down Taylor with a red sticky note. Okay, so very quickly. So everybody indicated some familiarity with BLAST. And I'm going to assume that, so what we're going to be working with today, just because it's simpler than working with next-gen sequencing data, which is what I normally do, we're going to work with BLAST. And my experience is that everybody in biology who's done much of the way of genomics or gene analysis or whatever is familiar with the concept of BLAST at the web interface. Just say you go to the NCBI BLAST website or some other website and you put in the gene you're interested in, pick BLAST, and then you get back a list of matches to that gene. Okay. And we're not going to use BLAST that way. We're going to use BLAST at the command line. And we'll talk about some of the advantages of that. One of which is that you can then use R to interpret the results of large-scale BLAST. So BLAST is a sort of canonical example of database search in bioinformatics. You have some query, and you may have more than one. And you have some database that you're searching for matches to that query. And the search can be either a nucleotide space, if you have a nucleotide query in a nucleotide database. It can be in protein space. If you have a protein query in a protein database, that's what we're going to be doing today. And it can be in translated. It can be translated in various ways. So you can do a nucleotide query in a protein database, or a protein query in a nucleotide database if you want to search a genome for a gene, or what have you. So this is BLASTn, BLASTp, BLASTx, tBLASTn, all those. We're going to be doing protein to database. And we're actually going to be building our own database, which I'll talk about. Because we're going to be, what the sort of, our job today, and it's always hard to pick a bioinformatics example for a mixed class of people who don't necessarily have the same research agenda. Our job today is to look at mouse and zebrafish homology, and then mouse and cow gene morphology. It's just a problem that I happen to have available, so we're just picking it up. And what we're going to be doing, let's see. So what BLAST does is called local alignment. So you have your query, and you have your matches, which we're going to call S1, S2, and S3. And BLAST can find local bits of your query that don't globally match to the subject, right? So different parts of the matches may match to the query. And this is why it's called a local alignment. You may actually have different portions of the subject, just to really draw a confused mess. You may have different portions of the match sequence matched to different portions of the query. If there's been an insertion or a deletion or a domain swap or something else go on that's biological, one assumes. And so this in particular is why it's called local alignment. It doesn't insist on a single unique global alignment. It can find many different matches to many different portions of your query. And the matches are represented as an alignment, which you may all have seen. We'll get a chance to see them in text format. And then That's all I'm going to say about basic BLAST. Now, BLAST can be used for a lot of things. And the thing that we're going to be using it for today is making assumptions, finding putative homologs. What I mean by putative homologs is situations where we have a gene query and we're asking from this gene in one species what genes there are that match it in another species. I don't know how many people here are sort of evolutionarily interested or engaged, but basically the terms I'm going to use, and I will do my best to just use homologs, but sometimes other words may creep in. Okay, I'm going to try and write that properly. The two words I'm going to use other than homologs are paralogs, and orthologs. And homologs just mean two genes if two genes are homologs, they have a common ancestral gene. That is the same gene in cow and mouse. Maybe a homolog if at some point in the past there was a common ancestor of mouse and cow, and that gene was present in that genome, and then the genome diverged, and mice and cows resulted, and now we're looking at that particular gene in mice and cows. That's an ortholog. That is a specific version of a homolog called an ortholog, which is the genes are related due to speciation. Paralogs happen due to duplication. So both of these are homologs. Paralogs mean the gene duplicated within the genome. So you can have paralogs of a gene in mouse. So I don't know, the Hox genes are example of paralog. The Hox genes are within the genome are examples of paralogs, and the Hox gene, Hox, I don't know, that's an example of Hox 1A would be in two different vertebrates would be an example of ones. The reason why I'm going through this is partly because we put some stuff about this in the text, and the other reason is that there's an assumption that many people bring in and that they use BLAST for, which is that genes that are homologs have similar molecular function. Right? So if there's a transcription factor in one species and there's a gene that matches that you know what it does, it's human or mouse, it's a lot of experiments. And you find that gene in another species and it has a high similarity, you go, it's probably a transcription factor. And maybe it's even doing the same thing. And that's a tricky business, right? Because like, as you may have noticed, mice and cows don't particularly look alike in some ways. They actually look an awful lot alike when they're very small, but anyway. So homology is, so there's this logical chain, which is that I'm gonna align, I'm not even gonna talk about it past this, but I'm just warning you. So that when you talk to a bioinformatician and you say something like this, or even worse, you talk to an evolutionary biologist and you say something like this and they go, no, stop. I now believe you know nothing about biology. You can say, oh, Titus did warn us about this. Here's what I actually meant. So we're going to do a typical bioinformatics thing, which is we're going to say sequence similarity implies homology, which implies shared function. And this is what we're after. This is the evolutionary statement, and this is now a bioinformatics statement. And this is a chain of logic that doesn't always, or even necessarily often, hold true. Now, as you'll see, for some of the genes we're looking at genes that are essentially like 80 to 90% identical at the amino acid level. So I feel pretty confident they probably have a shared molecular function. Just because they have a shared molecular function doesn't mean they do the same thing, right? Transcription factors get repurposed for many things. So this is the last I'm going to say about this for now. But I just want you to be aware, we're using sequence similarity, which is a computational observation, to infer evolutionary similarity, which is a statement about evolutionary history, to infer molecular function, which is now an actual molecular statement. And each of those links is potentially problematic. So just, you've been warned. Great. Anything I missed? Okay. This is a very, 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 very common workflow. Right, in biology. Like I found a gene, I wonder what it does. I found a gene in my nifty new organism, I wonder what it does. Or I found a putative gene in my nifty well-known organism and I want to know what it does in other species. So you blast it and then you go read the literature and then you come back and say, this gene is involved in cancer. And then you read more of the literature and you're like, wow, every gene's involved in cancer. All separate set of problems. And there's a couple of good, anyway, there's a whole bunch of biology stuff that I can talk about now that I will try to avoid. But that's what we're all really interested in, right? We're interested in the end biology. Okay, so how can we do something like take all of the mouse genes and all of the cow genes and find putative homologs or even putative orthologs? And that's what we're going to be talking about as a workflow. So I will erase this so that we have a nice, actually I guess I can, now it's too late, but I guess I can just put down the okay, so if we go to our RStudio and we make sure that when we hit enter, can I do a plus on this? Well, if I do a plus, will it actually decrease the font size? Yes, okay, don't increase your font size and it's gonna sort of, it could mess some things up. But if you were hitting enter and you were getting nothing but repeating straight, that means your binder is alive and well, and life is good, okay? So we're not actually gonna use R to start with, we're gonna use the shell. How many of you have encountered the shell? Some guys previously. So almost everybody. It's fine, you're not gonna require much knowledge about the shell, just know that it's a way to run commands on the computer. So what we're gonna do is start by going up to Tools, and we're going to click on Shell. And what that does is it gives you this green thing. You will have a different string here. You'll have a different set of letters here, but you should have a blinking cursor. And we're going to type a command. And the command is going to be echo, single quote, echo PS1, all caps, equals, single quote, $ space, single quote. That's actually in the materials. In the first set of materials, the link at the top. Here you can see it's word wrap, it's line wrapping. And what this is going to do is it's going to take this prompt and it's going to turn it into a $. And we're doing that solely. Okay, I don't know, I got that totally wrong. We want to do PS1 equals single quote $ space. Okay, there we are. We don't need the echo. And this is now going to be our shell prompt. And we just decreased the size of this. It's just some magic to say, make this long prompt into a small short one. And then just for good measure, let's tell the computer to say hello world. I have 2 R's because my keyboard's acting. Anyway, so this is a command prompt where we can tell this computer to do stuff. I believe all our studios that are running on Mac and Linux computers will have the same tools shell thing to run. There are other ways of getting to the shell. That involve terminal programs or SSH or other things, and we're just using it all inside of RStudio because it's just simpler to all be in one interface. So that's the other thing I want to say. Where are we running? So where we're running on some random computer. We're all running on some random computer that's running on Google's compute infrastructure, and I don't know where it's running, and we don't need to care. No, it's running in the cloud. Everything I'm going to tell you today will work for you on most on Unix computers that you have access to. And what I mean by that is generally Linux computers like the farm or the Genome Center cluster or a workstation or an Amazon computer or a Jetstream computer or anywhere where you would normally be expected to do data analysis. I'm using this because we could run probably half to 2/3 of what we're going to do today on your laptop, but then we'd spend the 3 hours installing the stuff. So for a half-day workshop, we tend to use a pre-installed environment. I can tell you exactly how to get this environment working. I have instructions. But for now, I just want to say there's a magic button. You click on the magic button. It spins up some computer in Google somewhere and redirects you to this interface. And that's how we're just going to do things today. So, okay. It's a 2-day workshop. We tend to do the software install. And we do have help available if you have trouble installing some software. Taylor helps run 2 different weekly help sessions, the Davis R users group and Meet and Analyze Data, where you can come and get help with software install. Is that a fair? Yeah. And we'll talk a little bit more about that. Okay, so what are we going to do here? So what we're going to do is we go back to HackMD and click on Running Command Line Blast. And this is now a tutorial. And what we've done is we've gone in our studio, we've opened a shell by going to Tools and Shell. We've made the window a little bit bigger, and then we've typed this in the prompt to reconfigure the prompt. So the first thing we need to do in order to run Blast, the command line, is we need to get some data. And we're going to get 2 mouse data sets and 1 zebrafish data set. These are actually data sets from NCBI. This is I don't remember if it's all predicted mouse proteins or some. We can actually find that out. And these are the protein coding sequences from mouse as published by the NCBI. We have to get them from a different location for this because of some technical stuff where they blocked NCBI's website on this computer for other reasons. Not me. What this does is this downloads 3 files, and we'll look at the files in a second. But the first thing is always to find your data. And you can get these kinds of files for essentially any published and annotated genome. You can get the list of proteins. So you could do this for 2 microbes. You could do this for. So what you can do is copy. I would highly recommend copying, and then go over to the RStudio and paste, and hit enter a few times. And what it's going to do is it's going to go and download these files. And the download should be really fast. And then over here, you'll see the files appear, or you should see the files appear in the file. So window, you can see we downloaded 3 files, a 12 megabyte file, a 3.9 megabyte file, and a 13.3 megabyte file. Yeah. Okay. So these files are now sitting on the remote server, on this remote Unix machine where we're running these commands. And everything we're going to do is going to be on this remote computer. And when we leave for the day, everything that's on that remote computer is going to go away. And you can use this window if there's something that you care about. You can use this window to actually download the things. You can use a checkbox and go to More and Export and download this to your laptop. Everything we're going to do though today should be repeatable by you tomorrow without me standing up here, because all the materials are available and everything. So you don't need to save like the general stuff that's in the tutorial. It's everything. Oh, copy paste. Shift insert. Okay. I think you can also do edit and paste. So you can also go up to edit and paste to do the downloads. I will say, for those of you that are suffering through Jetstream in the GGG201B class, we could be doing everything in RStudio or everything in Jupyter Notebook if we wanted to, but getting to the point where you can run this involves going through the painful part that we're already going through. So I've just opted for keeping the pain. I would welcome your feedback after this tutorial if you think maybe we should just be switching over to this instead. Okay, so we have these files. You need some? The paste or the?
The paste. So it's copy, right? And then press Shift Insert.
Okay.
What's insert?
So a lot of the commands in the tutorial are built for doing things at the command line, not using RStudio. So you can do an ls-l, which lists the files in the directory. And if you type that, if you type ls-l, you should see essentially the same information that's in the file viewer window, but you'll see it in this sort of text format. And you can see mouse.1, mouse.2. And please, again, feel free to flag me down and just ask a question if you have a random question. But hopefully we'll answer most of the questions. Okay, so all 3 of the files that we downloaded are what are called FASTA protein files. That's what the FAA extension sort of means. And they're compressed with gzip. That's what the gzip means. So .FAA means protein FASTA file. .gz means compressed with gzip. And we're going to uncompress them next so that we can sort of look at them and work with them. The reason they're compressed is because we're downloading them over the internet, so we want that to go fast. Now that we have them on this computer, they're not that big, so working with them uncompressed is fine. And the way you uncompress them is you type gunzip star.faa.gz. That says is use the program gunzip and tell it to do its work on any file that starts with anything and ends with .faa.gz. And now over here in the file viewer, you should see, how do we refresh this? There we are. You should see if you refresh with this little there, you should see the files are now bigger because we've uncompressed them. It's 50 megabytes, 8 megabytes, and 40 megabytes. All good? Okay. So what's in these files? We're going to look at the first few sequences in the file using this command head. Okay. And this is ugly on my screen, so I'm going to do the other thing we'll see if I can do this actually. So I'm going to click on the file. Okay. It's too big to open. So this is a perfect demonstration of why we're using head. Our studio is like, please don't open a 50 megabyte file. Okay. But head is like, oh, if you just want to look at the first 10 lines of this file, that's fine. I'll show it to you. Okay, so let me just, how do I get rid of this? This? There, okay. And I'm going to increase this a little bit and rerun head. There we are. That's a little prettier. Okay. I'm assuming many of you have seen a FASTA file before, but this is essentially what all of our genomic information is distributed in, is text files in this kind of simple format that you could, if you really wanted to open a 50 megabyte file, you could open in Word and look at. I don't recommend it, but you could. And the file format consists of what's called a FASTA header line, which is a line that starts with a greater than, has an accession number for NCBI, has some description, and then a species, Mus musculus domesticus. And so you could go look this up in NCBI, and you should see exactly this sequence associated with all the other links. And then the way the FASTA files work is the FASTA file, this 50 megabyte file, consists of many, many records, each FASTA record header line with a bunch of protein coding sequence, amino acids under it. So this is how all the genes in mouse are represented in a 50 megabyte, 58 megabytes of text. What you think about it is, I'm just going to say is approximately right. I'm going to make up some numbers. Let's say 20,000 genes. Let's say each gene is about 1,000 amino acids long. That would be about 20 megabytes. Yeah, 20,000 genes times 1,000 characters is about 20 megabytes. So order of magnitude, it's 20 megabytes, 50 megabytes. I'm going to declare victory. Some genes are longer. Sometimes there may be alternate protein isoforms present in the file that are redundant. In fact, that's one reason why the file might be bigger. I'm also not taking into account the size of the names in that. But that's what this file contains. It's just, I believe now, the entire mouse proteome. Okay, and the reason I like working with mouse is most of the genes have a name. It's not like some janky. I really harsh on plants a lot in GGG201B because their genomes are a mess. It's not like some janky plant or microbe that you just sequenced and you're like, look at all the genes. Yeah, this is what Taylor does. Look at all the genes. They're not named. They're named putative gene X. And you're like, what does that do? And I'm like, I don't know. Let's do a blast. And then the rest of this workflow starts to make more sense. Okay, so what are we going to do with this? What are we going to do with this gene, with this set of genes? We're going to blast all of them. But here's another thing that I've helpfully erased all the workflow stuff, but one of the things I really want to suggest that you avoid when you're working with a large amount of data is not to work with all the data while you're developing out your workflow. Right? Imagine that there's a step that's going to take 2 days. Don't run the step and then only realize at the very end that you got a parameter wrong, that you're not finding anything because you told BLAST this was nucleotide and it's actually protein. And so BLAST found all the wrong things or vice versa. So generally what we do is we subset, when we're developing the workflow, we subset the data. So what we're going to do here is we're going to take the first 2 sequences from this file, and we're going to put them in a new file called mmfirst.fn. So run that command. Okay. And now magically we get a new file over here, which is called, where is it? And then first.fa in the bottom, and you can now open this because it's kind of small. Can you open it? Does it not know how to open it? Why can't I open it? It's opening it up here. Why isn't it opening? Is it because it's an FA file and we don't know how to open it? Okay. Viewer, no.
No, it should open next to the script, but.
Okay, I've done horrible things to my RStudio. We'll come back to this. Let's not worry about it for now. What we're going to do instead of opening it is, okay, now what horrible things have I done? Okay, that works. This doesn't work anymore. There we are. Okay. So we've created this file, and now we're going to do, let's look at it, and I'm going to use the command cat. And what cat does is unlike, so head just takes the first 10 lines, cat displays the whole file. So here we have two complete genes, the NADH dehydrogenase subunit 1 and NADH dehydrogenase subunit 2. Why this order? Anybody guess? There's no reason. It doesn't matter. In the proteome files, the order it's in the file doesn't matter. Just some arbitrary database order that NCBI has. They don't sort it by meaningful gene. There's no way to do that anyway. But the other thing is, how do I know why did I use? So I used this command head -11 to create this file. Why did I use 11? Any guesses?
11 lines.
Right. So that gets 11 lines. Why 11 lines? So head by default does 10 lines. If I did head -10, oops, mouse.1.protein.fa, I get this. That's the first 10 lines. Why 11?
So you don't truncate the second gene.
So I don't truncate the second gene. Okay. So absolutely. And then head, what did I call it? And then first.fa. How do I know that I truncated the gene before and I'm not truncating the gene now? This is dumb, detail-oriented bioinformatics stuff.
Play around with the head until you figure out how many lines it is.
But so I'm asking the question you just skipped over, which is how do I know that in this case I'm not truncating? Because it doesn't. This line isn't 80 characters long.
I would also check to see.
Right. If you did a head 12, you should see the next header line. And so I just want to say, this is going to sound kind of dumb and kind of silly. So I do head -12. You can see I pick up the first line of the next gene. This is all going to sound kind of dumb and kind of silly. Like surely in this age where we have, sending spaceships up, like we could have our program just give us the first two genes. And the answer is welcome to bioinformatics. Like the amount of sort of manual crud that you have to do with text files and be worried about getting the details right is still very extreme.
For the redirection sign, is that a command made only for head, or is it made for other default things
So that is an excellent question. The question is this greater than sign is a redirection sign that says take the output of head and put it in this file. And it works for any program that prints stuff out. So you can take any, and we'll actually use this, we won't use it for BLAST, but this will work for any program that prints stuff out, except for the ones that it doesn't work on, which is like some of them. So, but a lot of the Unix programs, it works. So what I'm doing is I'm taking the first two sequences because what I want to do is get BLAST on those first two sequences working. Once I have BLAST on those first two sequences working, then I can go for broke and do BLAST on a lot more sequences. So let's get BLAST on these two sequences working. And the first thing I need to do is make a custom database. What I mean by making a custom database is BLAST, remember, it searches with a query against a database of sequences. That database needs to be organized. It may consist of multiple files. It may take in multiple files and all of that. And so what we need to do is tell BLAST what that database is and what type of sequence it is. So here we're going to do make BLAST-DB. We're taking his input zebrafish1.protein.fa, and we're telling make BLAST-DB that this is a protein database. If you get this wrong, if you put in, I think it's nucl, N-U-C-L, then there will be problems down the road because it'll be like I'm trying to search a protein sequences with nucleotide sequences, and I don't understand what's going on, and look at some weird errors. And so one of the purposes of that head command that we used before, just to look at the top of the file, is it amino acid sequence or is it nucleotide sequence? You can tell very easily by looking at the file. You'd think BLAST could, but anyway, I don't know why, but they require that you tell them. And now we're going to do our BLAST search. So run this. Oh, I forgot to run the make BLAST DB. Sorry. Okay, so run the make BLAST DB. That's important. And that actually creates a whole pile of other files over here. And then we're going to do BLAST p-query. I'll put this up at the top. And what this does, and it output a lot of stuff. What this does is it takes those two proteins that we put in mmFirst.fa, and it searches them against all of the zebrafish genes. Okay. So first of all, let's just take a step back. How cool is that, right? Like in like a second, we're searching all of the zebrafish proteins for these two mouse proteins. And what gets even cooler is we can do it with 50,000 mouse genes if we wanted to, without caring. We just have to wait a little bit longer, maybe like 50,000 seconds. So one, but whatever. Computer time is cheap. So there's a whole bunch of output here. And what I'm going to do is make use of our studio to scroll up. And you should just be able to scroll up using the normal scroll button. And I'm actually going to make this bigger again. I'm going to rerun it, scroll up. And let's go, I'm going to go all the way to the top, which is the very top, which should be this. Yeah. And so here you see a bunch of stuff like how to cite this stuff and reference and whatever. The next output that you see from BLAST is the database we used. It has 53,000 sequences, 38 million total letters. And then there's the first query. And so all the web BLAST stuff that you do is a wrapper around this program, in effect. We're just using the program sort of, when you go to the website and you run BLAST, the output that you get is basically this output, but it's like been reformatted to look pretty. But this is what it's running underneath. And so we have our query, which is this NADH dehydrogenase. And then we find one significant alignment in zebrafish. So it turns out zebrafish also has NADH dehydrogenase. Excellent. Subunit 1 in particular. So I'm guessing from this is actually a mitochondrial gene. Seems reasonable. And the first match, it has a bit score of 426 and an p-value of 10 to the minus 150, which is pretty good. So the bit score we'll get to later, the e-value tells you the expectation of finding this gene by chance, of finding this kind of match by chance in a database of this size. So you would have to do 10 to the 150 times many searches in order to get a match this good by default. There's one major by chance. There's one major problem with that, which is there's a lot of assumptions that go into that e-value. And one of the major assumptions that go into that e-value is that proteins aren't related to each other. It turns out proteins are often related to a lot of other proteins. So this is really a background calculation. This says nothing at all. Like you could be using, if you search for the zinc finger gene, where there's just a lot of homologs around, you're going to get a lot of matches that are sometimes very specific and that aren't even remotely homology. What's the word? That aren't actual. That are at some, many hundreds of millions of years or billions of years evolutionarily related, but they're not the homologs or paralogs you're looking for. And so you have to worry about things like conserved domains and other things. BLAST takes care of a lot of the sort of low complexity sequence that might just match because a string of purines looks like a string of purines, but that it sort of takes care of that with this compositional matrix adjust. But so this e-value, the sort of rule of thumb is an e-value depending on what you're doing. An e-value like this is very good. Like this is a good match. You get an e-value of like 10 to the minus 3, and you're searching with a weird random organism that's a microbe that's very distant from everything else. That might still be real. It just depends on the biology of what you're. And then we get to the cheddar, the really cool stuff, which is the match. So here you see the actual alignment. And I don't want to go too much into BLAST because, whoops, because there's actually like whole books written about what it does and how it works and how to interpret it. And I'll just say that this is an alignment. The query sequence from position 4 to 63 lines up with the subject sequence from position 10 to 69. Where there's a letter, the alignment is exact. Where there's a plus, this is a conservative alignment, and these two amino acids have similar biochemical properties. So standard BLAST. And where there's no match, it's just there's been some sort of substitution. And you can see, like, if you get runs like this is pretty much the same gene, right? It aligns across. This is pretty much guaranteed to be the same gene. It aligns across the whole distance. Mouse and zebrafish are about, depends on who you talk to, they're about 350 to 400 million years apart. Last I remember, if you get a match like this, okay, it's mitochondria, so you'd expect a good match because otherwise things die. But you know, this is good across that evolutionary distance. If you're comparing mouse and human, a lot of your matches are going to look like this because we're both mammals. Mice are like 100 million years divergent. So on protein evolution in vertebrates, it's slow. The reason I'm saying all of this is, first of all, I'm validating myself as somewhat of a biologist of some kind to you all, and the other is that this all matters, right? If you're looking at, if you're searching for if you're working with a plant that's quite divergent or a fungus that's quite divergent, you're not going to get good matches. So you have to do more work to sort of look at them. If you search with two organisms that are essentially identical, one's a strain variant of another, or there's a word that people use, the variant, it's cultivar, I don't know, cultivar, right? You're going to get basically identical genes, and that's okay. But you just have to interpret what you're seeing in the context of the biology of your search. Okay, so we did this BLAST. We did 2 query genes against all of zebrafish we found. For this first one, this is NADH subunit 1, NADH dehydrogenase subunit 2. Subunit 2 looks almost as good, but we get 3 matches. Subunit 2 matches to subunit 2, subunit 5, and subunit 4. The other 2 matches are relatively poor. If we look at the first match, you'll see that it does not look as awesome as the first one. Right? There's not as much block. There's not as many blocks. It's still very good statistically. If you look at the second match, which is down here, you'll see that there's probably what's going on here is that there are 1 or 2 or maybe 3 domains that are conserved between them. That's an inference. I don't know anything at all about this protein. One of the fun things about doing bioinformatics is, you just go swimming in the sea of all the genomes, and you're like, I don't know any biology at all, as it turns out, basically. Because you find weird stuff, and then you have to read up on a bunch of literature, and that's why many of you are in grad school. So anyway, so this is trace similarity, but it's 10 to the minus 6 e-value. So it's much less rare. It's a much higher e-value than previous one, which means it's more likely that you would see this match at random. So you can already see something, or I'm foreshadowing something that's going to come up in a few minutes, which is, okay, great, we inspected 2 of these, and we reached some sort of conclusion, like we can use an e-value cutoff, we can get rid of these other 2 values, we can do this for more genes, but once you get past like 20 or 30 genes, this becomes a problem. Right? It's like, okay, well now I have literally 30 pages of text to read through. And you could do things like print out the text and take a highlighter and be like, I believe this one and I don't believe this one, and so on and so forth. And I'm going to show you a different way. It's not necessarily a better way, but it is maybe a more time-convenient way of exploring these results. And that is to use the magic of R. Okay, so we've run BLAST. It looks like we're getting reasonable matches. That looks like what you'd expect out of vertebrate things that match in a line, yay. What do we do next? Let's do, well, the first thing we can do is we can build a text file out of this so we don't have to scroll up and down. So to run this command, and I'll run it and then I'll explain the command. What this command does is it's the same command as before, but we've added a dash out, which says save the results to this .txt file. And this is now something that is over here. Should be over, I've lost my windows too big. There's a refresh somewhere in here.
At the bottom.
That's the bottom. mmFirst.xzebrafish.text. And I still can't open this because
You should be able to without selecting it, only clicking on the.
only clicking on this. I assure you I'm clicking.
Mine opens.
Well, la-di-da. I did something weird in my, I'll have to figure it out over the break.
Yeah, it's opening up here.
You can pick it. It's just if it's a 50 megabyte base file, like half a gig, then you don't have to it. Otherwise, it's.
Okay, well, so I don't know why that's not opening, but anyway. So you know what I'm gonna do? I'm actually gonna download it. I'm gonna export it to my laptop, and then I can open it here. You'll see it's a text file. And so this is now something that if you wanted to, you could actually open up in Word, you could highlight things, you could convey it to other people, you could make notes on it, whatever you wanted to do, right? This is a text file that's now on your laptop if you did that. What I did was I selected it, I went to More, and I went to Export, and that let me download it to my laptop. So this is an antidote to the, I don't want to like scroll around in a terminal to look at my laptop. Okay, but you still face the problem that if you do this for like, say, all of the mouse genes against all of the zebrafish genes, you're gonna have 50,000 blast output, which is annoying. So how do we deal with that? Well, let's do more sequences. Let's take the first 500 lines of the mouse protein and put it in mmsecond.fa. And then we're going to run a blast. You can copy and paste both commands at once. We're going to run the blast. And that's going to take a little while. It's probably going to take like 10 or 20 seconds. So, sorry, go ahead. Really? Okay. So, but that's okay. We have time because I can talk more or less endlessly. So, so a couple things. So one thing is I believe I verified that this actually stops at a gene boundary. The other is you'll notice a couple things. I'm naming the files differently. Why am I naming them mmFirst and mmSecond? Well, I'm more or less picking those names out of a hat. MM stands for mus musculus. First is just the first file we're using today. Second is the second file we're using today. You could name them in a more meaningful way, but for a workshop where we're sort of making up our own problems. So you could name this like MM top, MM more, if you want. It doesn't really matter. The other thing is this file name is actually kind of interesting. For the output file name, I'm saying name it mm-second.xzebrafish.txt. Why am I naming it that? Any thoughts?
Descriptive.
It's descriptive, right? So in the fairly common event that in 6 months, I'm like, okay, I want to go back and look at suppose this file takes 5 hours to build. And so I leave it sitting on my laptop. And then in 6 months, I've had a meeting with my advisor, and my advisor's like, hey, I want you to go back and look at this gene, or these 30 genes. You can either go back to what you ran and rerun it and regenerate these files, or you could go to your directory and go, hey, you know what, I bet this is actually the file I want to look at. So you, 6 months down the road, is the consumer of these names. And you might be thinking, erroneously, that your memory's great, and this is all wonderful, and you're going to remember this stuff. And I have a freakishly good memory for file names. And what I will tell you is you may come back to this file in three years after you've moved on to a postdoc job or a faculty position. You've gone through a bunch of life events, and your memory is now shot. And this is the situation I find myself in all too frequently. I literally spend probably 10 hours over the last week recovering a bunch of stuff, figuring out what I spent a couple of weeks doing last year so that Taylor could figure out could repeat what I had done. And if I just luckily I named the files not super horribly so I could figure it out. But like I spend a lot of my time resurrecting old projects so that I can rerun the things now. So naming things is really good. This is also not only descriptive, but it tells me, if you have a consistent naming scheme, it tells me this is mouse versus zebrafish at a glance. And it's a text file, which tells me that I can open it or something.
How did you determine that maybe they cut off the gene?
So if you run this without the greater than, it will, let me see what happens if I run it. Okay, so it's still running. So no, it's done. So if I do head minus 500 of mouse.1.protein.faa, you can see that I get, did I get it wrong? I might have gotten it wrong. I think this is actually, I think there's probably a missing part of the chain. So I lied. I just made 500 up. But what you could do is you could say, oh, well, 500 gets me these 2 extra lines. So if I back off and do 498, I'll just get this conclusion. But realistically, at this point, what I would probably do if I weren't in a workshop is run all of the mouse genes against all of the zebrafish genes, and then I wouldn't need to worry about subsampling the file. So what does this file look like? Well, I'm going to use a command line way to look at it, which is this less command. Oops. Hello. What was that? Cool. All right. I'm going to use this less command, and this is a command line way of looking at the file that lets me page down. So it puts me at a prompt, and I can hit the space bar to page down, and I can see, maybe I can hit the space bar to page down. I can't actually do anything at the moment. Am I the only, hopefully I'm the only one having these problems. Q should get you out of it. Did yours freeze? Excellent. All right. Well, so maybe we should be going over here and doing it over there. Anyway.
But I mean, you can just toggle down with the up and down keys, right? I mean, that's how mine is working. If it doesn't freeze, it's still really good.
Yeah, mine is freezing, which is a problem. Let me do less mouse second. What was it called? It was mm. Got to put too many m's in. It's really hard to read from over here. Okay, so this just freezes my. That's really odd. Why does this freeze my RStudio? Okay, I don't know what's going on, but if I were to download this file over here, maybe less doesn't work in the RStudio console.
Well, it worked and then froze.
Yeah, so don't run that command.
Oh,
You can exit out of your terminal and
If you want to, you can click on the little X up here and restart the terminal by going up to Tools and Shell. You can actually have, you should be able to have multiple shells running actually. You do like a new terminal. And now I have, okay, that didn't do what I thought it would do. Anyway, but all of that aside, what we now get is a much bigger file that has a lot of BLAST results. And this highlights the problem that we're going to deal with next after the break, which is how do we deal with interpreting this?
if you use the back arrow, you'll scroll back to your first terminal.
Oh, look at that. Okay. Okay. So we're going to do one more BLAST before we're going to take a break in just a minute. We're going to do one more BLAST. And what we're going to do is we're going to make BLAST output something that's readable by R. So it's going to be something that's not human readable per se. It's basically a comma separated or a tab separated value file. And we're going to do that by specifying out format 6. You can see all the possible BLAST output formats, but we're going to run this. And it's going to take as long as the original BLAST. We're doing the same amount of computing, but we're just outputting in a different format that we can load into R. And while that runs, I would also like to ask you to do one other thing. So I'm just going to wait a second. Looks like everybody's got their hands off their keyboard. If you go up to File and you do New File, and you go over to R Markdown, and you click on it, it will ask you. We'll circle a little bit. Any second now. And it will say, we need to update things if we want to use R Markdown. You click on Yes, and this will take about 10 minutes, which is why it's a great time to then take a break.
Since we already created a text file, can we just transfer the text file into an article?
So, so the question was, since we already created the text file with the BLAST, couldn't we have just moved it into a version that's parsable by R?
Yeah.
And the answer is basically no. there's a couple reasons for that. I actually spent many years doing exactly that. But it turns out that the default blast output is really meant for humans to read, and so they do things with placement of numbers that is somewhat arbitrary and changes frequently. So getting it into a spreadsheet format that RStudio can read is just a whole different thing. So it's an interesting exercise. You know, what I will say is a good trick is to do your big blast with a way that's readable by R, so that you can look through it and get a grasp of what's going on, and then go back and cherry pick like 10 genes at random and do the human readable blast just to sort of check the alignments and stuff. Or use the web interface at that point. I mean, if you're only doing 10 genes, I would just use the web interface because it gives you links to all the links and stuff. Okay, so as I said, this is going to take about 10 minutes. And so why don't we reconvene at 10 after 11. Bathrooms, water, coffee. And we're happy to take questions and stuff while you're, I'll just be sticking around here. Actually, I'm going to go get water and then I'll be back. And then I'll be happy to take questions. If you can find a way to have your laptop not go to sleep, that would be ideal.
Yeah.
Oh, no.
Type less, this is one. Solution 2 is the inactive denomination. And mine started actually with this computer. My phone has at the bottom, I would pop my hands up to do process.
Oh, sure. I just wanted you to help stuff as well. I guess you're giving you to do it.
Yeah, we're gonna need to okay.
Sorry. This is what I wanted. We used it every year before that.
Will we use binder as coming here instead of starting at your battery? Right, but just for the blast.
You could do it. You could have it as a backup for.
Are we using gesture?
Well, as a three, so there's the suffering.
Go grab my orange.
Chocolate. But I also do it. What e-value range do you usually use as like a cutoff? Or just one of those
So
where it depends?
It totally depends.
Are you
The minimum e-value that I would consider for looking at, so just specialized is all 10 times 3. It's going to be called 10 times 3 is going to be hard to make sense of that. So you'll still find for really divergent things, like if you're looking at weird viruses, still find legitimate things to do with that. But then I would use something like a markup model and use structural information.
Yeah.
Last. You know, and after that, it totally depends, right? Some genes are fast moving, fast evolving.
Yeah.
So 10 to the minus 12 is my sort of default cutoff, but this is probably real homology, with the exception of things like zinc fingers and other things, right?
Right? That's the problem we're trying to solve.
And the real trick to publishing that kind of thing is you just find somebody else that's used it and you say, as in blah, 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 we use this cutoff as an initial filter. Otherwise, your reviewers are like, how would you pick your p-value?
Yeah, I don't know. I would have like a instruction for how to download like R somewhere.
Yeah,
So,
so
part of my.
the data carpentry instructions are in science applications. Can you find those?
Yeah, I was just wondering.
Like, I'm not solving it.
Yeah.
I mean, like, it's gonna be more practical stuff, just the style. Yeah. That's
Yeah, so if you follow these setup instructions here, so that link is datacarpentry.org/r-ecology-lesson. I'll put that on the link in D.
So, I mean, regardless of all the science going on. That's okay, Sarah.
And you're
Helen,
Helen said top.
yeah, side,
Sarah,
Sarah.
Kuzai.
The odd thing is I know your email addresses. It's fine, anyway. Great. What's up?
I don't know, regardless of all the cool science going on, I really like your teaching style, and I was wondering, I guess, like, how did you, like, I don't, right, part of my grad studies is gonna work on science education.
Yeah.
And the way you set up your tutorials is a lot like elementary school teachers set up lesson plans. I was wondering, like, how did you come across that, or like, how did you develop?
So I have no, it's okay.
oh, yeah. Oh, actually, I don't know if I have your email, but.
Or it doesn't
All right. So if the R Markdown thing completed, you should have a window like this. And you can just click cancel because the important bit was we installed it all. We're not actually going to do anything with it just yet. Okay. So assuming that this worked, and you remember you can hit enter a few times just to make sure that your RStudio's live. Is it?
It's still frozen on that previous command.
Oh, try clicking X on the terminal and then starting a new shell by going to tools and shell. Good.
So, sorry, you said hit cancel on the right?
Yes, you can click cancel. So just to explain what went on, we went to start up some new functionality, this R Markdown thing, and because we're going to use it later, and RStudio was like, oh, you don't have the right software installed, despite me having the right software installed, but never mind. And so then it installed the software, but we don't actually want to open a new R Markdown file yet. That's for later. So you can click cancel now.
Status code 404 is timeout.
Status code 404 is timeout. Yeah. So you need to start up a new binder.
And rerun all of them.
Yeah.
But just the, I mean, okay. I think I
Just the
when I
last.
got it.
Okay.
I got it.
Okay. So start up a new binder and see if you can run the. I need to make this file downloadable. Sorry, I didn't.
Is that a new terminal? Sorry, new terminal.
You'll need to go back to the binder, the little binder box that was in the HackMD.
Sure.
Which is here, and click on this launch binder. And I'm actually just going to take one quick second. I'm going to make this file that we just generated. I'm going to put it up somewhere so that we can download it without rebuilding it. So let me just figure out where it is. Second. Zebrafish.tsv. Wait, what's going on? Let me just do this on my laptop.
Okay, so where are you at?
I launched a new binder.
Great, so then run the.
So I'm going to make it so it's downloadable.
Okay.
The TSV file that was generated was downloadable. So hang on a sec.
So in a second, download the file that he makes downloadable.
I can do that. Cool.
Yeah, so that's the buggy part of our studios. So what do you do? Yep. There. There. Okay. And then let's see it through the sections. Go all the way down. Session. Sorry.
So I'm just going to put down at the bottom on the website, I'll show you in a second, I'm going to put a command so that if you had to restart your binder, you could just download one file to move on to the next stuff. If you needed to start RStudio, you will need to re-download this file.
And that those instructions are in the HackMD?
They will be.
Okay. So once this relaunches, you'll have download instructions in HackMD to get them on out. Did anybody else have time out?
Okay, So let me see if I got this right. If you go to the HackMD, on the bottom of the HackMD, there's a thing where if you needed just to restart our studio, you'll need to re-download this file like so. And this should work to recreate the TSV file that was the last thing that we tried to generate. Okay. And at this point, I want to say one other quick thing, which is this is going to be a quick advertisement. If you need a place to host files for either collaborators or for everybody in general, and they're under 5 gigabytes in size, there's something called osf.io, which is the Open Science Framework. And it's where I am hosting these files in a public place. But I can make this private. I can have this be private, and I can share this with collaborators. And it's a way to distribute fairly large files, not raw read files, but like files, variant VCF files or RNA-seq calculations. And it's free, so you can go sign up. And so you can actually, if it's, I can show you more of this one-on-one or later, but you can actually go and see all of the files that we use today are uploaded here. And so I'm making them available that way. And it's basically better than Dropbox for a couple of reasons, one of which is that they're all versioned, and the other is that this is actually, when you publish a paper, you can actually include this as supplementary material. This can be a place where you've placed your materials for the paper and made them available, because you can freeze this, you can archive this for publication purposes. I'm happy to talk more about that. It's sort of a side note, but it's called osf.io. Interesting. So I'll actually put that into the notes here. I'll just say, if you are looking for file storage and publishing options, check out osf.io. Okay. So, but back to this. Okay, we now have this file, which is mm2nd.xzebrafish.tsv. It's a megabyte in size. It's 1,000 kilobytes in size. So it's pretty big. So what are we going to do with it? What we're going to do with it is we're going to load it into R, and we're going to do some plotting. And this is, I kid you not, this is literally how I do most of my bioinformatics. Is I generate some file and like, I wonder if that worked. And then I load it in and I graph it and I look at the data and I go, okay, this actually makes sense. Or I go, this doesn't make any sense at all, given what I expected. And so I try and just sort of characterize the file. And usually the first thing I do is I look at the top of a file. For a TSV or a CSV file, that's not always so useful because it's sort of ugly. And what I'm going to show you in R is actually a pretty good way to do it. So we're going to move on to visualizing blast score distributions in RStudio. And now we are not going to be using the shell anymore. The shell prompt here in the terminal window is what we use to sort of do the big computation, the slow computation. Now we're going to move back over to the console, which for R is where you actually type R commands. And I'm going to be a bad person. I'm not going to teach you to start up an R script and put these commands in. Should I? Should I be doing that? Is that bad? Is it okay to teach at the console? Okay, that's clearly a no. So let's try this. You've all indicated that you're familiar. It's entertaining. So Taylor's a grad student in my lab, right? So she's sort of occasionally slightly hesitant to tell me I'm full of it. And that's what you're seeing going on there. There is a correct way to do it. Titus is not saying it, but anyway. So what we're going to do is we're going to go up to the source window, and you should have something like untitled in there by default. And what we're going to do is we're going to copy and paste things from the lessons into here, and then we'll run them from here. And I'll go through that again. Most of you indicated you had some familiarity with RStudio, so we'll go through it again. But essentially what we're doing is we're writing a script, and then we're going to run the script in R. One line at a time. So the first command we're going to run is not that command, is this command, which I'll go over here. And this command is an R command to read in this tab-separated file basically saying it's a tab separated file, so the separator is a tab, and read it into an R variable called blast out. So I put this in this untitled thing, and the first thing I'm going to do is name it something. I'm going to click on save, and I'm going to name it blast, just blast. And so now this is a file that if I want to remember the commands I ran, I can download from RStudio, and it'll be on my laptop, and I could upload it later on and come back and run these commands. So just put up a red stick if you need help getting somewhere. So the thing is, I didn't actually run this command, right? I put it in an R script. Let's run it. So, and Taylor can tell me if I've got this right. But basically, I'm going to go up to it, and I'm going to click on run. And what run does is it takes it from this file, and it runs the line that I was on down here in the console window. And that now actually does something, part one of which is it creates a new data frame called blast out. You can think of a data frame as basically a spreadsheet in R. You can actually come over here, and you can see this data frame has 14,000 observations of 12 variables, which basically means it's got 14,000 rows and 12 columns. And we'll talk about what those columns and stuff are in a second. So we've loaded in the output. So one very quick thing is, this is pretty cool, right? This is a megabyte of data. This is 14,000 lines, and it took R, like a second to load it. So, yay. Okay. So if you now run view blast.out, and this you can actually run directly down here in the console, you'll get a nice view of the spreadsheet here. And the only problem with this view is we didn't have any column headers in this TSV file. Blast didn't give us any column headers in the TSV file. So these columns are all named not so useful things like V1 and V2. So what we're going to do is we're going to name the columns like so. We're going to actually, first let me go back to blast.r. So here what I'm doing is I'm saying name the columns of blast out this set of things. And if I run that, now when I switch back over to this tab view of blast out, you'll see I've named the columns all of these things. And so here, queries, what you're seeing is the columns are now query sequence ID and subject sequence ID. So this is the query, and these are matches to it. Are people more or less okay with where we are? Sorry, I sort of skipped around a lot of tabs very quickly. So there's the file that we're editing, and then there's the view of the data, and then there's the console where we're actually running stuff. Now, if you look at this, you might be going, where did these column names come from? It turns out these column names actually come from the BLAST documentation, and you can go look at that to see what they are. But we're only going to be concerned with, I think, with a few things. One is going to be the percent identity. Another is going to be the bit score here, which is basically how many amino acids line up. And then the last is going to be the e-value, which is that expectation score that we saw before.
So you have to add the headers in because that's not in the regular.
It's not in the TSV file that BLAST creates. If it were, R, I think, would figure it out. But I'll ask Taylor, which is stuff. I'm pretty sure that if there's headers in the thing, it will figure it out.
Okay.
And there may be, you might have to say use the headers.
It's artisanal, but yes, that is true for the most part. With read.table, it assumes there's no headers. With read.csv, it assumes there are headers. But you can explicitly code that into your command.
So you can modify read.table to say use headers because they're there. In this case, we knew there weren't headers because the first line was consisted of like the query sequence ID, the accession number, so we knew those weren't the headers. That's why you look at your data. Yeah.
It looks like whichever line you're at and you press run, that's on one button.
Yes.
Run at.
So very good point. When you're running things in RStudio, if you're over here, you put the command, you put the prompt, the cursor on one line, and you click run. It only runs that one line down in the console. If you want to run all of them, I believe you can do, and again, Taylor will have more to say, but you can select all of them and run them both, and it will say it runs blast out and it runs call names. And probably that's what this does, but I'm not 100% sure. Probably, but I don't know.
What was the question?
How do you clear this console?
I
Okay, Control L might work. Control L works. And that actually works the shell as well. And you might be thinking, how possibly does Titus remember this stuff? And then I would say, I've been doing it since 1988. And that's how that's it.
Is there a good resource for those commands, just the basic like commands that are useful?
So there are a couple good resources. There's a manual. There's actually manuals in here. So you can actually go here and you say, read, if you want to know what, I think we used read.table, right? Read.table, and it'll actually tell you all of the different options. That can be a little bit overwhelming, right?
Okay.
Like, oh my God. So what I would do is I would go to Google and I would say, how do I read a TSV file into R and assign column headers? And then I would probably end up at Stack Overflow, which is a help site where you can post a question and then multiple people can answer the question. And in theory, the best answer gets upvoted. And so, I have this table. I need to read the table in. And then there's all this other stuff, and it's how should I read it? And the first answer is what you needed to do is specify separator. See, it's perfect.
Yep.
You specify the separator as a tab, and there are headers in the file. And this is how everybody I know actually learns this stuff. You basically spend a lot of time Googling.
Okay.
And then at some point you spend a little less time Googling because you've already done it like 100 times. And then 20 years later, people start asking you.
Then you teach it.
Right. So that's basically, yeah. Nobody can remember all this stuff. The other thing I'll do is I'll use my own teaching materials because I don't use R a lot. Yeah. If I need to know how to do something in R, I'll go back to the lesson that I'm giving you now. So this is whole scam, right? Basically what I'm doing is I'm writing teaching materials that I then go use and tell my students to use so they don't have to remember the commands. So I just sort of have arranged my life around this kind of convenience. We can argue about whether it's truly convenient, but anyway, so that's basically how I look at it. There are books, so like Vince Buffalo has a book, Bioinformatics Data Skills. There's Practical Computing for Biologists, but realistically, I find those are great. If you have a week to sit down and really work through the book, that's great. But Stack Overflow is where you're going to end up as soon as you have a question that's not answered in the book. Okay, so what are we going to do? So there's a couple commands you can run. We already know a lot of this information. That this is has 14,000 lines and 12 variables. I just want to show you some of these commands so that if you're writing an R script, you can print this stuff out without having to run it in RStudio. There's a command called dim, which stands for dimensions, and this is the same as this. It basically tells you how many rows and how many columns a data frame has. You can also do head. Whoops. You could do head of blast out, and what head will do is it will do exactly what head does at the shelf, but here it's going to print out, and it's ugly because my screen's so large. But what it's going to do is it's going to print out the first 10 lines of this. You could also simply go do that and view it here. The only reason you would use things like head and dim down here is if you're writing a script. It was a long-running script at the command line, and you wanted to sort of have output, because this stuff won't be available to you if you're running the script at the command line. So it's good to sort of have this sort of dual thing in mind. One is I'm writing this interactively. I'm doing this interactively to develop the script, and then at some point I'm going to say, well, now I want a script that's going to find all of my homologs. It's going to pick the e-value cut off intelligently, and it's going to do some plots. And I want that to run the command line, so I don't have to start up RStudio to run it, and on big data sets I don't have to run it. And then you use commands like din and head to make sure the data looks right when you're running the script outside of RStudio. Plus, I think it's a rite of passage. You have to show people them in R, whether or not you're using RStudio. So let's do a plot. So I'm going to run this command hist. Hist blast out $e-value. So what this command is doing is it's saying build a histogram of the column e-value for the data frame blast out. And this is where the value that it's useful to have the names of the columns in the spreadsheet, because now you can refer to them that way as opposed to V1 and V5, which is not so useful. So if we run that, and we can actually put this in blast.r. How do you run this whole thing? Do you have to just do select all and run?
No, there's keyboard shortcut.
Okay, well, there's a keyboard shortcut. Okay, well, anyway. So, and then the graph shows up over here. Okay, so what's the standout feature of this graph?
Very high to the left.
There's a bunch of zeros, basically, right? And what does that mean for e-values? There's a bunch of very low e-values, which means there's a bunch of pretty good sequences. Is that a reasonable, there's a bunch of pretty good blast matches. Is that a reasonable conclusion from this data set? So you're nodding, and this is a trap. It is not. Because e-values, so one thing to notice is look at the scale of this. It's 0 to 10. Well, first of all, I'm just going to tell you I know this, because I know this, but blast's maximum e-value is 10, and what an e-value of 10 would mean is that by default I would expect 10 matches at random to the sequence. So the e-values we're really interested in are all down buried in the 0 to 1 bin. So how do we get at them? So you could be clever and know more R than I know off the top of my head and set this axis to be logarithmic. Or you can actually transform the column values itself by specifying the transformation. And so what we're going to do is we can actually do a hist of the minus log of the e-value like this. So I'm going to take this and I'm going to do hist of minus log blast out e-value. And you don't have to remember the plus or the minus because you'll look at the graph and be like, I want that to be flipped, so I'm going to put a minus there. Like it doesn't. And so what that's going to do is it's going to log transform the e-values, which is actually the appropriate thing. Now it's going to say, well, an e-value of minus 140 is 140 in this. An e-value of minus e to the minus 3 is going to be 3. So when we do this and we run it, I don't actually know how to run it from the
Command enter for one line, you're just going to parentheses now.
Stop it. Okay, command enter. So if I hold on a Mac, if I hold down command and hit return, it runs it automatically. This looks more interesting. Right? This now tells me something that if I had looked at the data more, I would know. Just there's a range of really good e-values, e to the minus 130, e to the minus 140, e to the minus 200. And there's a range of kind of crappy e-values of, greater than 1, e-values greater than 1 turn into things that are less than 0 here, or e-values of around 1 to minus 1 to 1 times 10 to minus 10, or minus 10, right? And this is the kind of thing, sort of trying to be philosophical a little bit. This is the kind of thing you want to see if you haven't done any filtering or selection of your data, right? This is like, well, we should expect some garbage. We should expect some good stuff. And the goal of your exploratory data analysis is to start assigning cutoffs. And you can do this in a totally half-assed way, which is typically how I do a lot of bioinformatics, which is to say, well, there's like the really good stuff, there's the kind of good stuff, and there's a pile of junk. So the first cutoff that I would consider is probably going to be around e to the minus 20, just because it's a dip. I mean, this is totally phenomenological nonsense. But it actually sort of fits the fact that this is biology and we don't, like statistics, at this level, we don't really know what the right e-value is. And we should actually be going and looking at some of these matches to figure out what they are. And we can do that, and we'll get to the point where we can do that. Okay, so does this sort of make sense as what we're doing? This is characterization of the data. Okay, and now I'm going to, okay, so we can also do this with another column like bit score. And bit score actually, in some ways, looks kind of similar to the e-value to the minus the log of the e-value distribution, which is not by chance, as it turns out. If you actually stretched this out, it would look an awful lot like the e-value distribution in terms of its shape. As it is, you have to sort of know that it should be the same because it's not spread out enough. How do I change the y scale? The x scale, sorry.
xlim.
So I can say xlim.
That would be inside of hist. So it's an argument to hist.
Xlim.
Is equal to, and then you do c.
Right.
And then the two numbers.
So let's say 0, 400. Okay, and now this is stretching it out. It doesn't give me any more bins though. How would I? This is great. This is programming by grad student. How do you get more bins?
The cheating way is if you go back up to line 6 and add a comma.
Line 6, yeah.
And then after that, and then press tab.
Oh, tab. And it tells me
all sorts of
different options. So I went up there, I put a comma in, I hit tab. And I don't know, it's not.
Breaks is the one that you want.
Is it breaks? Oh, breaks. Wow. How about this? A single number giving the number of cells for the histogram. So this is why the internal RStudio help is like good if you know what you're looking for.
Yeah.
Breaks in. Let's get fine grained. Let's get something fairly fine grained. Did that work?
I will say if this doesn't work, it's because I always use ggplot.
Okay, well, it's not nice. Sorry. Okay, that didn't seem to work.
Try density instead. Not on top of.
The density of shading lines?
No, I'll figure it out.
Frequency. Representation of frequency. Yeah, I don't know. So I don't know how to do this. Anyway, I don't know how to stretch out the axis. But, well, actually, so how do I increase the number of bins in rstats for hist? And choose bin sizes, understanding. So now the problem is I've encountered somebody who's clearly gamed the search, and I'm finding a company that's so this might not be so useful.
You have to have R after usually.
I put rstats. So the other problem with the R language is they named it R. And it turns out to be extremely hard to Google for R. Interesting factoid. One of the reasons why Python is better than R is because Python was named after Monty Python.
So
Basics of histograms. Exact number of bins.
that's the answer right there.
This one?
If you look at it, use the breaks argument. Hist beta breaks is equal to
Seek
1, 0 to 80.
start to the end and then the iteration. Okay, so I'll do this just because now it's a point of personal honor to get the graph looking right. It's something like breaks equals, what was it? Seek.
And then we have 0 to 400. And then I believe that's an L. Yeah, L is equal to 20.
Not span range of X. Okay, so I'm going to give up on this as just a bad gig all around. Okay, we have other fish to fry. So here's the histogram of bit score, and so what are we looking for? So I'm going to tell you that the bit score is related to the number. It's related to the number of amino acids that align. So a higher bit score means more amino acids align between each query and each match. So higher one is better. It means you have a more strict alignment. And so if I wanted to pick only proteins that had between 250 and 300 amino acids that align, which is a lot, I could select only those rows that had the bit score that was this big, and I'll show you how to do that at the end. So why would I want to do that? Well, okay. So why would I want to pick out these and not these? Just from a biology perspective. Because we're trying to find homology. These are pretty likely to be evolutionarily related. These are fairly likely, and then these are probably less so, right? You only have 10 or 15 amino acids. So it's all about filtering the data and exploring that filter. So I'm going to sort of drive this into the ground, this point into the ground, and let's do another kind of plot. So is there a relationship between bit score and p-ident? So p-ident is percent identity, and bit score is something that's a slight bit fuzzier, which is how many amino acids line up. Would you expect there to be a relationship between those two things? How many sequences line up and what their percent identity, how much of the sequence lines up and the percent identity between the sequences? You would generally expect there to be a relationship. I can diagram this out if anybody is interested, but the plot is kind of interesting. Okay, so it's a pretty ugly plot, I apologize. The main thing to note is there's something that's kind of linear-ish here, and then there's a bunch of garbage down here. So what's going on? Well, it turns out that you can have, so it turns out that we're running into one big problem here. And this is where like understanding your data is important, which is percent identity is length normalized. So if you have a really long gene and only 5% of it is aligned, you're going to have a p-ident down at 5. But the bit score, if it's a really long gene, that 5% could be a couple hundred amino acids. So you're running into the problem that, at least one problem, which is that, that these are on different scales because bit score doesn't account for gene length and p-ident does. And so that's the value of using things like e-values and percent identities and so on, is that they normalize for features of the data that affect the significance of your results. So how can we see that? Well, this is my favorite kind of trick, which is you do what I would call slightly BS bioinformatics hypothesis testing, which is you keep on plotting relationships until you find ones that make sense graphically, and then you're like, I think I understand my data now. So here what we're going to do is we're going to multiply the percent identity times the length of the query, of the match in the query, which is the if you remember the blast output, this is the end of the match in the query minus the start of the match in the query, which is the length that's matched. And we're going to plot that directly against bit score. And now we should see a more linear relationship. And let's see if we do. Put this in. And this now actually looks kind of like a line. So there's other things going on here. In particular, bit score has a slightly different relationship with those conservative amino acid changes. So bit score takes into account that some amino acids are identical and some amino acids are conservatively similar, right? Whereas bit score does that, but percent identity doesn't. I believe percent identity, just if it's not identical, it discards it, right? So that's why there's sort of, it's not a perfect line. But this is pretty cool. This tells us something about how BLAST works. This tells us something about how bit score works. This tells us something about how we can filter things. In particular, once you can get this relationship out, it tells you that you can use bit score as kind of a proxy for quality of match. And that's kind of where you want to be with this. You sort of want to vary the parameters and explore the relationships in the data set. And I just want to say, like, the value for me is in the visualization. Like, I really like seeing the data and looking at the rows. This is 14,000 rows of data plotted, and you've picked out a nice linear relationship, and you can actually do a fit to this and pick out what the relationship is and do other things. And I think probably the main difference in the relationship here is that I believe percent identity is in percent, not in fraction. So we could even modify this further if you like playing with your data, and you could say, I'm going to take p-ident and I'm going to divide by 100, and I'm going to multiply by the length. And now all of a sudden, this is starting to look a lot more like a one-to-one line. Anyway, so you can sort of play with normalization features and so on and so forth. This is my favorite kind of data analysis, because I feel like I've learned something at the end of the day. So this is really an example of what I call initial exploratory data analysis, where we poke around with the data to see roughly what it looks like. And we're not doing statistical analysis. We're not trying to confirm a hypothesis. This is really generating sort of our initial. And I would just say, as a philosophical point, typically if you have small replicate sizes, so if you're doing 2 or 3 replicates, which is pretty typical in RNA-seq analysis and so on, often a lot of your bioinformatics is going to be what I call hypothesis generation. You're going to be trying to extract signal from the data to then verify some other way, maybe doing more targeted experiments than doing qPCR or something like that. And the reason for that is that given the volume of data and the noise that's typical in these large-scale experiments, if you have a fairly low number of replicates, it's actually very hard to conclude something statistically about that. Now, there's a bunch of papers on that I can, depending on what kinds of things you're doing. Does that make sense as a statement? Often, faculty that are less used to dealing with these large data sets will be like, well, we paid an awful lot of money for this data, so you should be able to conclude something from it. It's like, no, we're just trying to get good hypotheses out of it so that we can move on to the next step. And it depends on the field as to whether that's publishable or not. Sometimes it's publishable, sometimes it's not, depending on. Okay, so some quick questions. Why are we using R for this instead of the Unix command line or Excel for the exploration of the tab separated file? Any thoughts?
If you are trying to publish, you probably can't publish with Excel.
So that is probably not accurate in most fields. Like you can publish Excel analyses just fine, I think, in many fields. Doesn't really matter. You can't in yours, but it's kind of cool. Okay. But what I would say is Excel, you're dealing with 14,000 lines. Excel probably has no problem with that, but if you were doing all of the mouse against all of the zebrafish, you'd have probably maybe 1 million or several 100,000 lines, and Excel might be less good at that. R will have basically no problem with data sets that are even 10 to 100 times this size. So that's one reason to use this. The other is, and I'm not doing a very good job of showing you interesting plotting, and that would be a whole other tutorial, but you could make this plot publication quality with relatively little effort. And now you've generated it using a script, and for your next data set, you can generate your same publication quality thing using the script, which you can just download. And that's pretty cool, because now you and your undergraduates and your lab mates, and if you want to become a professor, your students can just use the script to look at this kind of data. And it's also automated. So everything we did, there's like essentially 4 commands to run the blast and generate this figure. You can just do it very quickly. And you can actually, if you did any interesting statistical cutoffs, you could put that on the paper. So if you did anything interesting statistics, you could say, this is my R script, here's what I did. Feel free, and here's my data. Feel free to rerun it if you're a reviewer. Okay. Yeah, the reason we're not doing it at the Unix command line is basically the Unix command line is for running commands on files. It's not for interactive visualization and exploration of data. So that's the dichotomy in that workflow is there's some stuff that I'm running that takes a long time and that takes in big files and outputs smaller files, whatever. And that's best done at the Unix command line. And then the sort of interpretation of the data and visualization of the data is done in R or in Python with a notebook. So since we're somewhat short on time, what I actually want to do is show you something that I think is super useful. We're going to go to the bottom of this next tutorial, which is exploring Schmalast orthology. And I'm going to show you how to subselect. So something that we want to do, so just to motivate this, I want to know what this point is. How do I find out what that point is? Well, right now, you don't really have an option. You can scroll through 14,000 lines. You could sort this so that you can find things that have a high bit score, You can look at things that have high bit scores. But suppose you were interested in what's in this bunch of, what's in this blob of data, the blob of the data. Well, for that, you actually need to do something that does sub-selection. So at the bottom of this, which we're not going to go through, the very bottom, it recapitulates a lot of the points above. Schmalast is actually something that does the orthology assignment. So what we were doing before was BLAST, where we were trying to find homologs. And we're just saying, find me any match to this query, and then we're going to apply cutoffs later. What Schmalast will do is we'll actually do blasts in both directions. It will search mouse against zebrafish, and it's essentially doing mouse against zebrafish and zebrafish against mouse, and then asking where do those match? Where does the same gene hit in both directions? It's called reciprocal best hit, and it's a fairly common way in bioinformatics of finding gene-to-gene matches across organisms. So that's all what's up above. And what I wanted to show you is at the bottom, there are two interesting commands. And what we're going to do is replace Schmalast sub with blast sub and Schmalast out with blast out. And what we're going to do for this first one is we're going to randomly subsample the data. So head is great in that it shows you the first 10 lines. If you have a big data set, the first 10 lines are unlikely to be in any way representative of the whole data set. If instead we randomly pick 10 lines or 100 lines and look at those, maybe we can see other things. So this is a useful sort of look at my data technique. So I will rewrite the command and then copy and paste into the hackmd, but you can replace Schmalast with blast. Did you have a question? Okay. So I'm going to take this and I'm going to replace last, and blast. I'll just go put this over on the hackmd in case people want to just copy and paste. Actually, let me put it over here. Randomly subsample 10 rows from blast out. And so this is now down at the bottom. It's this command. So if we do that, what we do is we create a new variable, blast sub, which now has only 10 observations. And if we run capital view blast sub, we can get a nice view up here. And we can see, so the first thing is the subsampling took the data frame headers along with it. And now rather than the first 10 things in the tab separated file, now we have 10 randomly selected. So there's different sequence IDs. And you can see that it's a little hard to see because my screen's so big, but you can look at them and see if they make sense. And so one thing you could actually do, you could say, gosh, I have an e-value of 4 times 10 to the minus 88 here. Let me go look at these two genes. And just because I like to show that this actually works, if I go to NCBI, actually in Google now you can actually just search for the accession number, which I think is pretty cool. Okay, I didn't copy it, right? Let's copy it. So I can Google for it and it doesn't match. Okay, well that's a little sad. It usually works. Okay, well I'd have to go to GenBank to search for this. See, every unplanned excursion. Just a bad idea. Okay, nucleotide is the wrong thing. Or if it'll tell me if it's a protein. Okay, so this is anyway, gastrolysing finger protein. I could look at the other one. I could see if they're the same. Right. So these identifiers that we have in our studio, that we have in our spreadsheet, are literally things you can go look up in the biological databases, which is pretty cool. But the other thing we want to do is we kind of want to look at specific bits of that spread, specific bits of that plot. So this is a way to subset that data frame. So in this case, Schmalast out, but it can be blast out. We can pick query lengths. We can pick long genes or genes between 8,000 bases and 11,000 bases with low bit scores, and we can ask questions of them. But we can also put in any other kind of sub-selection we want. So let's develop this. I'm going to take this code and bring it over here. And then what we're going to do is develop something, why is my copy paste not working? Copy. And paste. So the first thing I have to do is make this blast out. And then let's look at this plot. What's a point on this plot that we're interested in? You know what, I'll pick this one because it's like standout easy. Right. And I'll just say, okay, I want things that have very high bit scores. So I'm going to take all the rows that are above, let's just take this one. So this is probably what, this is like 400? So this is probably like 800? So let's just pick out all the points that have a bit score above 600 and see what happens. So to do that, I'm going to modify this so that bit score is greater than, what did I say, 600? And now if I look at temp by typing capital view temp, there's two genes that have bit scores of greater than 600. Let me copy this command first. There's two genes that have a bit score greater than 600. That two matches that have that, and you can see that the bit score matches that, and I can go identify what these genes are. And to do that, how would I do that? I'm just going to go do the GenBank search to find out what they are. And maybe this will work. Cytochrome C oxidase subunit. That's probably going to be something that's pretty highly conserved. I believe it does something biologically important. I don't really remember much of my biochemistry, but right, essential for life, that kind of thing. So that's a good sign, right? So this isn't a specific and kind of crafted example of how you should interact with your data. Poke at it with a stick, find a few data points that either stand out or that are right in the middle, go look at them, do the sort of biological due diligence of looking at what the gene names are and seeing if they make sense. So I could look at the next thing I could do, right, is this is the match in zebrafish, right? This was zebrafish. Now this was the match in mouse. So if I come over here and I take the zebrafish one, it should also be cytochrome C oxidase subunit. I don't know why we've had such a mitochondrial focus, but anyway, it is running. And yay, Maniorario is the zebrafish. This is right. So this is the right gene. So what we're saying is for these long bit scores, at least in a couple cases, we have matches. So I will tell you, as somebody who sits on a lot of qualifying exams and reads a lot of theses and reviews a lot of papers, that at least for your qualifying exam and thesis defense, or thesis committee, I guess you don't have defense, we don't have defenses here so much, but when some mean bioinformatics professor says, how do you know your 50,000 rows of results are correct? If your response is, I went and I looked at 50 of them in detail, and 49 of them were correct, and the last one was an artifact that I understand, that's a good answer. Because the chances, if you picked at random 50 rows from your spreadsheet and looked at 50 of them and 49 of them were correct, the odds are pretty good that you have like a 95% correct spreadsheet. Yeah, it's a little hand wavy, but you can't look at all 50,000 of them. That's unreasonable. And so what you do is you do a random subsample, you look at a few, you understand them, and then you know. And the odds are the first 3 times you do this, like, wow, I messed up. I am selecting the wrong genes. I got the cutoff wrong. I did something wrong. And then at some point you'll get used to it and you'll have the cutoffs right, and then you can move on with the rest of your analysis. The same thing when, like we talked about in the GGD class, if you're doing variant calling, and you look at the alignment of your reads against your reference genome for each of 10 variants, and you don't see a pile of reads lined up with that variant in them, then probably you got your variant calling code wrong. That works whether you have 10 variants or 1,000 variants. You can just look at 10 of them and see. Right? So looking at your data is a really important aspect of this. Okay, so I hope, so the last thing I wanted to talk about just for like 10 or 15 minutes was R Markdown. And then you will be free to escape with a good conscience. Are there any questions at this point about sort of the workflow that we went through, running things in the command line, loading them into R, and then exploring them in R? Okay. I will say, if you are interested in saving this file, depending on what you put in there, you can go over to Files. You can go down to wherever blast.r is, and you could do More, Export, and Download, and that will now download it to your laptop. And then later on, you could go click Upload to upload this saved file. So this is not how we recommend running RStudio or doing your data analysis, but if you ever are in a situation where you don't have RStudio installed, you can actually come back to this. You can actually come back to this website, start up a binder, upload your R file and your data files, and do your analysis here. And the really great thing about it is that this is something you can give to collaborators. If you don't want to have to make them install stuff, they can actually run the same thing because it just uses a web browser, and it's free. So the last thing I want to talk about is sort of documenting your workflow. This is the R Markdown section. And so what R Markdown is it's a way of taking these graphs and putting the code and the graphs and a description and a discussion about the code and the graphs all in one place. This may sound to the naive it's surprisingly similar to a publication, to a paper. And the main reason it's not is because we're not 5 years from now. In 5 years, I actually expect, this is a aspirational statement, but in 5 years, I actually expect that a lot of the time you'll be able to write your papers in this kind of notebook format, because a paper basically is some text, some figures, and some citations. There's no reason at all that we couldn't be writing them in RStudio. And in fact, let's be honest, a lot of people do write papers in R Markdown already. And that's what I'm going to show you right now. But I don't want to make it intimidating or anything. What we're going to do is we're going to start up a new file. We're going to copy some of these commands over to that new file, and then we're going to run that new file, and we can produce either an HTML or a PDF from that new file. So the new file we're going to start with is this R Markdown file. So now go up to File, New File, and R Markdown. And now, for those of you that went through that install step before, it should just pop up. If it pops up and asks you to install stuff, you can go through with that, but it's going to take like 5 minutes, so I'm sorry. And the default output format is going to be HTML, which just means a web page. You click on, so give it a title like Blast, make it you the author, and click on OK. So I'll say Blast, and the author will be Titus, and I'll say OK. And what we end up with is an untitled file, untitled, and it has some sort of metadata at the top, which is the title, the author, the date, and the output, and then a bunch of other stuff. And we're going to do two things with this. The first thing we're going to do is I'm just going to delete everything below. Should I not? Should I leave it?
There was just one little code block that's really friendly to keep there.
Okay, we're gonna. That first one is really friendly to keep there. So what we're gonna do is delete everything from here on down. You don't have to do this. Don't worry about it if you don't want to delete stuff. If you don't want to make.
You can also delete lines 12 through 14.
Right, but I want those lines there.
Oh, so wait.
Because here I can put my favorite Blast thingy, and I can say some other text with emphasis. And this is a language called Markdown. It's actually what HackMD uses behind the scenes. It's fairly common now in both RStudio and Jupyter notebooks as a documentation format. And basically, it's an alternative to Word, where you don't have to like select something and then say italicize it. You just put a star around it. Star, italics, or bold. I should really know that.
You would know.
Yeah. Anyway, we'll see. We'll see how it turns out.
Italics.
And this is how you do titles. And so the cool thing about this is a single document that can include R code, text formatted for human consumption, and also the plots and stuff that are output by R code. So that's what we're going to do. We're going to do, so before we do this, we're also going to save it because saving things is good. And I'm going to name it Blast Doc. Doesn't matter what you name it. I'm going to name it Blast Doc. So what we basically need to do is put in some commands, some R commands. So let's come over here where we have a whole file full of R commands, yay, that we know work. And I'm just going to copy these first 3. So let's make it the first 4 R commands. And it doesn't really matter what you put in. You can copy them off of the lesson too. They don't need to be from that window. And I'm going to enclose them in its back quotes, right? 3 back quotes, space, 3 back quotes. And do I need to put R in here?
Yeah.
R and that's it, right?
That's all that's required.
Okay, what should I be putting in?
A title for your book.
Did you see I interpreted the tone of her voice.
So
Sorry.
you put a space, and then you can name your code chunk. So like.
Load into plot.
That's great. And then you see the chunk hops up down there, right at the bottom where it says.
Chunk 2, load in plot.
Yes.
Okay.
So it's a great way to navigate your chunks.
And the thing I'm going to do is I'm going to get rid of this intermediate hist because this was the wrong hist that showed us that not the relationship. This is the plot that I want to get Instagram on. Okay. So, and let me just, you know what I'm going to do? I'm just going to copy this whole thing. I'm going to come over to the HackMD, and I'm just going to put this in the HackMD as my whole blast out file. And you can now see in the HackMD at the bottom, this is the, whoops, why didn't that select the whole thing? Seriously. My keyboard is not being shown at the moment. Oh no, it did copy the whole thing. It just has that. This is going to be hard because it has triple back quotes in it. Okay, well, here's the code block. That's the important bit. All right. So this is the code block here. And now, coming back over to our studio, so now, save this, saved, and click knit. And what knit will do is it will turn this into beautiful formatted HTML. And in my case, it says I couldn't get a pop-up window to go, and then you just say try again, and then it works. And then if we've done all our things right, if we've done all of our, whoops, if we've got all the code right, we end up with something like this. And this is now a beautiful exposition of, okay, the plot's a little bigger, but beautiful exposition of the code I ran and the output of the code that I ran, all in one document that I can turn into a discussion, right, of what's going on. So I could rewrite the entire tutorial in this format with the R code in there showing the plots in there. So what's the value of this? Any thoughts? Why am I bothering?
So you can go back.
So you can go back and figure out what you did, right? So I'll say one thing, which is that this plot is actually what happens when you run this code. It's not like there's a PDF where you like generate a plot in Excel and export it in PDF and then like put it in the Word document and then you changed what was in the Excel, but you forgot to update the plot. Not that that ever happens to anybody, but you know, that's a problem. The other thing is this is quite portable. You still need the raw data file somewhere. So, but if you have the raw data file and you have this RMD file, not the HTML, but the file that you built this from. That's something that somebody else can use to rerun your analysis. And then they can go in and modify these parameters. And they can do their own thing. And they can adjust analysis. And the most likely person to do that is you in 6 months when the reviewers get back to you with their comments about why they don't believe your paper and why you should use different statistical cutoffs and why the plot needs to be red, blue, and green rather than whatever else it is. And you can regenerate everything in one fell swoop. The other nice thing is you can send this to collaborators, you can send this to students, you can send this to your advisor. You can turn this into PDF and send it to your advisors so they don't have to learn R or R Markdown or anything like that, HTML, and it is nice, basically. So typically the way that typically what I would do if I were writing this up, this is exactly what I would. So okay, I'll give you what I view as it's not even aspirational anymore in my lab. A lot of cases now in my lab when we are doing our data set explorations, we do it like this. We either do it in a Python notebook or an R notebook, and this is what we share. And the fun thing about that is when we come across the same type of data set, but maybe from a different project, we can actually, I can take Taylor's code. Taylor has figured out how to do something tricky with taxonomic display or whatever. I can take her code, I can bring it over to the new project. I can immediately get all of the, as long as I can load the data in, I can immediately get all of the displays that she thought were informative. And even better, because I'm her boss, I can get her to help me with it. Anyway, but you know, literally I can start from the sort of expert position of somebody who's thought about this and struggled with code, and I can start from that position rather than doing it all on my own. And you can also include this as your supplementary material, as part of your supplementary material for a paper. Say, this is how I did my data analysis. And there's a subset of reviewers, and they'll mostly be the computational ones, that will say, okay, this person, it's like a signal that this person knows what they're doing. So it's a way to sort of, and it helps the reviewer because the reviewer can rerun everything you did, and they can see if you cheated, if you like sub-select the points in a particular way or whatever. Which, you know, if your goal is to publish, you know, robust science is actually a pretty good way to go. So that's really all I wanted to say. I would, you know, at this point, I don't actually know if it's easy to, can I convert this? If I knit to, oh, I can knit to Word. Let's try that. Okay, download file. What happens if I knit to Word and so this is like super advisor friendly, right? Look at that. There's my Word document. Right? This is now something that even the grumpiest advisor will accept. It's a Word document that they can mark up. So that's pretty cool.
Like testing, like if you're running statistics, can you show?
Yes. All the you can
just head, and then
you can include all the output, or you can include a summary of the output, or whatever you want.
Okay.
Yeah. It's totally limited by, it's only limited by what R will let you show. So you can head some number of columns, you can do subsetting, you can do plotting, whatever, and it will show up in here. Cool. And the only thing you have to watch out for is your advisor will come in with track changes and start changing your code around.
Just trying it to PDF yet?
I have not. You have to install other stuff.
I was going to say, does that ship with Jetstream or no?
Apparently it does not.
Binder.
But you can knit to PDF if you go through another 5-minute installation process.
It's less.
It failed?
Well, no, it's less trivial than the 5-minute installation process, and choose up 5 to 10 gigabytes of hard drive space.
Cool. It's a bad.
FYI.
Oh, because it uses LaTeX?
Yeah.
Yeah. So I'm in the rare position of telling you Word is a perfectly fine output format for this for the moment. But I mean, fundamentally what you're doing is you're generating the Word document or the PDF or the HTML from the blast, whatever I called it, blastdoc.rmd file. So all you really need is the raw data and the blastdoc.rmd, and you can regenerate this. So I could have shortcut this whole tutorial by saying, here's a black box of data that I'm not going to tell you how I generated, and here's my RMD file, and now you have this figure.
I have a dumb question.
Go ahead.
How do I I've always used R on my own computer. How do I export this out of
So
this?
with the RMD file.
Yeah, well, so
Yeah.
I want to export my RD and my R Markdown file.
So come over to the file browser over here. Select the file. So I'm going to select blastdoc.rmd. Got it?
Mhm.
And then go up to More and do Export. And then you can download it.
I have a question. Why is that first box that we didn't delete helpful?
What?
The first box, the R setup, why is that helpful?
Oh, yeah, line 10.
Yeah.
Titus, do you want me to?
Yeah.
Okay, so RStudio allows you a lot of fine-grained control over, or sorry, R Markdown allows you a lot of fine-grained control over what ends up happening when you knit your code. And so you see that the syntax of that is sort of annoying, where it's saying knit R, and then you have 2 colons, and then you're calling a function called option chunks. But to remember all of that is kind of frustrating. So I usually start my new document and leave that code there. And then in those little parentheses where you see echo equals true, that's where you put in all the fine-grained control. And if you press tab, you'll see how you can control those.
So you're answering one step beyond what I think the question was. Just like, what does this command actually do?
It makes it so that you can see the code in the output.
Right. So this echo equals true means in the Word document, for example, or the HTML, include the code, not just the text and the output of the code.
So if you don't want to include the code because your advisor has no idea about the code, you can just say false.
Echo equals false. And there's settings then also. Well, let's see what this does.
Yeah, let's see if this works. It's still on Word. Yeah, and now the code is.
And if you wanted the opposite, where you wanted the code in there but not the plot, you'd have echo equals true, and then include equals false. So there's a lot of these options, and you can go through and sort of explore what each of them do, or Google, or if there's some specific thing that you want, there's usually a setting for that.
And so when you look it up, you look at ops chunk and help. You look through the options there.
I would actually just recommend if you go back to that code block that we didn't delete, and you put your cursor where it is and press, put a comma and press tab. Those are all of your different options. And then Googling what those mean will probably be better, but some of them like warning and message or error, those ones are saying don't print out messages, don't print out errors, don't print out warnings, and then some of them are not intuitive. Like, what is DP? I have no idea. But those are all of your options.
Fig.width and other things are called. So anyway, so this is actually I have to say this is the first time I've tried to string these things together like this. It's actually a pretty comprehensive overview of how we do bioinformatics workflows. And it's how everybody else should do bioinformatics workflows, obviously. So, and more to the point, it's how actually a lot of people do bioinformatics workflows. So do the stuff at the command line, and then load it to R and interpret a display, and then record that into an R Markdown document that you can share with others. So, so as I said, this has all been recorded. I'll share the YouTube link with you. And if you have any feedback, please feel free to send me an email or chat with Karen or Taylor. And I guess that's the last thing I should say. So further learning opportunities at the bottom of the HackMD. There's a Davis R users group, which meets on
Mondays 10 to 12.
Mondays 10 to 12, super friendly. There's a meet and analyze data, which is actually held in this room on Thursdays 3 to 5 by my lab. Then there's this DIB training mailing list, which I think many of you are maybe on, where we announce this stuff. We haven't been super active because for other reasons, but we have this 2-week summer workshop for people that are interested in Illumina and other high throughput sequencing interpretation more generally. That we're running that was just announced, and we'll announce other things to that. There's the Data Science Initiative, which runs a lot of workshops. Could you add Data Science Initiative in there? And I think that's it. Generally, there's a whole bunch of data carpentry and software carpentry workshops that are like 2-day workshops. And those will generally be announced through one or all of these forums. So, and that's where you can learn more about version control and other things as well. So, thanks.
Thank you.
Thank you.
Thank you.